Good morning dear students, welcome you to our channel Bio by Gulab Sir. As a part of series of lectures, new chapter Respiration in Plants. Topic Glycolysis we are going to start today. Respiration is not a oxidation but it is a breakdown since it occurs in presence of as well as absence of oxygen. Therefore, instead of saying oxidation, it is correct to say that it is a breakdown of complex organic substance into simple organic or inorganic substance with the release of energy. So respiration occurs in both plants as well as animal. It occurs in every cell which is being a living. Without respiration or without this process, one cannot survive, right from amoeba to elephant or chlorella to banyan tree. So every cell, all the type of cell except anucleated RBC, that too except matured RBC. That is the point for NEET. Otherwise, glycolysis occurs in all the cells. What is glycolysis? It is a stepwise breakdown of glucose into pyruvate acid. This is the first part of the respiration. Second part of the respiration is Krebs cycle. So as to have complete release of energy, that is what we call 36 ATP bonding. You would be knowing that process of respiration. C6H12O6 plus 6O2 to produce 6 CO2 plus 6 H2O plus 36 ATP. That is a net ATP production. This process occurs in two major steps, that is first glycolysis and second Krebs cycle. So as to learn the first part of this process is glycolysis. Glycolysis is stepwise breakdown of glucose into pyruvate acid. Minimum there 10 steps are occurring in glycolysis. It occurs in cytoplasm of the cell. It occurs in all type of cell including bacteria, prokaryotic and eukaryotic. Since it occurs in cytoplasm, therefore need not to have the mitochondria. It occurs in presence of as well as absence of oxygen, hence it can be aerobic as well as anaerobic. But the ATP production is vary in aerobic glycolysis and anaerobic, and that is we are going to learn as a balance sheet of ATP. Now let us continue with the topic glycolysis as the first process. It is discovered and described by Embeden, Meyerhoff, Paranas. These are the three scientists, Embeden, Meyerhoff and Paranas. Therefore, this pathway is also called as EMP pathway. Glucose is the substrate of this pathway. This glucose is not readily present, but it is present in the form of sucrose inside the plant, and we, human being or animal, they have in the presence of glycogen which convert into glucose, and later it becomes the part of respiration, glycolysis. So in plant, sucrose is converted into glucose and fructose. And these glucose and fructose take part into the glycolytic pathway or become the substrate of the glycolytic pathway. So glucose and fructose are same. Glucose undergo phosphorylation to produce glucose 6-phosphate and then it undergo isomerization to produce fructose 6-phosphate. But fructose directly undergo phosphorylation to produce fructose 6-phosphate. Here, if glucose is a substrate, then glucose has to undergo isomerization, phosphorylation to produce the glucose 6-phosphate. And if fructose is suppose a substance, then directly fructose undergo phosphorylation to produce fructose 6-phosphate. Okay, it is clear. So let us continue with the step when glucose is the substrate. 9 steps are there. I read it: phosphorylation, isomerization, phosphorylation, cleavage, dehydrogenation, including this isomerization is the 10th one. 6. Dephosphorylation, isomerization, dehydration and dephosphorylation. This is the 9th and this is the 10th. These are the steps. Now how to explain? See, whatever we are saying, that is only we have to just construct the sentence. Glucose undergo phosphorylation. Glucose undergo phosphorylation to produce glucose 6-phosphate. How it undergo phosphorylation? Glucose combined with ATP to form glucose 6-phosphate in presence of enzyme hexokinase. How this step is written? Let you know. See here how I have written as a chemical reaction. See, this is the first step. Phosphorylation, glucose plus ATP in presence of hexokinase and Mg magnesium as a cofactor to produce glucose 6-phosphate. Second step, isomerization, same glucose 6-phosphate undergo isomerization to produce the fructose 6-phosphate. These are the steps what we have to write as a notes, and I have given that as a notes. Now let's come to the first page as we have to explain this process. So once this glucose 6-phosphate produce, it undergo isomerization in presence of isomerase to produce fructose 6-phosphate. Second step. Now third step, phosphorylation. Once again, how to write it in words? Fructose 6-phosphate undergo phosphorylation in presence of phosphate fructose kinase to produce fructose 1,6-diphosphate. Very simple. Fructose 6-phosphate undergo phosphorylation in presence of enzyme phosphate fructose kinase to produce fructose 1,6-diphosphate. Now, next step, cleavage. That means breakdown. This 6-carbon compound fructose 1,6-diphosphate undergo breakdown, that is cleavage, in presence of aldolase enzyme to produce dehydroxyacetone phosphate and phosphoglyceraldehyde, triose phosphate glyceraldehyde or phosphoglyceraldehyde. So these 3-carbon, 2 molecule, 1 molecule of DHAP, 3 carbon, another 1 molecule of PGL, 3 carbon, produced from this 6 carbon. So 6 carbon split into 2. 3 carbon, DHAP and PGL. They are isomer of each other. So they can convert in each other as per the requirement of the cell. When we require PGL, 2 molecule of PGL, DHAP convert into PGL and form 2 molecule of PGL. Thus, this 2 molecule of PGL undergo dehydrogenation and that is continued. But here we have to understand, since they are interconvertible, that is what isomerization occur in presence of phosphotriose isomerase. That enzyme also you have to remember. Now dehydrogenation. This phosphoglyceraldehyde undergo dehydrogenation. What does it mean? Hydrogen from phosphoglyceraldehyde release and combine with NAD to form NADS2. Whose dehydrogenation occur? PGLs. फॉस्फोग्लिसरॉल्डेहाइड का डिहाइड्रोजनेशन हुआ, उसमें से हाइड्रोजन निकला, किसके साथ कंबाइन हुआ? NAD के साथ. क्या बना? NADS2. That is called phosphodehydrogenation. Occur in presence of which enzyme? NAD dehydrogenase, nicotinamide adenosine dinucleotide dehydrogenase, or PGAL dehydrogenase, phosphoglyceraldehyde dehydrogenase. Why? Because phosphoglyceraldehyde has given hydrogen, that is dehydrogenation of PGL occur, therefore enzyme PGAL dehydrogenase. Ultimately what is being produced? 2 molecule of 1,3-DPGA from 2 molecule of PGA. I told you here you seems to be look like 1 PGL, but this converted to this when the process continue. Therefore 2 molecule of PGL undergo dehydrogenation to produce 2 molecule of 1,3-DPGA in presence of PGL dehydrogenase. I think this is clear to you up to this. Now this step is continued here because we have no space. So continue here. This 1,3-DPGA, 2 molecule of 1,3-DPGA undergo dephosphorylation. That means from DPGA, 1 phosphate release and combine with ATP to produce ATP. Here ATP are produced. And the byproduct is 2 molecule of 3-PGA, not 1,3-PGA, because phosphate from first carbon is released from that 2 molecule to combine with 2 molecule of ADP to produce 2 molecule of ATP, and thus the ultimate production is 2 molecule of 3-PGA. Now this 2 molecule of 3-PGA undergo isomerization. That means phosphate from third carbon is replaced to second carbon. That means 3-PGA become 2-PGA. Phosphate which was attached to the third carbon of PGA, it is transferred to the second carbon of PGA. Structural formula is changed, chemical formula is same. That means 2-PGA is isomer of 3-PGA. What is isomer or what are isomer? Whose structural formula is different but chemical formula is same. So here the same thing. So isomerization. So 2 molecule of 3-PGA undergo isomerization in presence of this mutase enzyme to produce the 2 molecule of 2-PGA. Now this 2 molecule of 2-PGA undergo dehydration, not dehydrogenation, dehydration, that is loss of water in presence of enzyme enolase to produce 2 molecule of phosphoenol pyruvate. These full forms you will get it when on the last page. Phosphoenol pyruvate. This phosphoenol pyruvate undergo dephosphorylation. So once again, 2 molecule of phosphate from 2 molecule of phosphoenol pyruvate release and combine with 2 molecule of ADP to produce 2 molecule of ATP, and thus the final product is pyruvic acid. Yes, here the glycolysis is completed. Although I have explained it in a first glance, so it was required to explain in a speed so as to have continuity of that reactions. You can go through step after step what I have been explained, and same thing I have written here. You can go see. These are the steps. This is the first step, second step, third, fourth, fifth, sixth, 7, 8, and last one is this 9th step, that is to produce the pyruvic acid. So this you can take it as a part of notes. Same thing I did explain and same thing is written over there. But at the last, what happened? How many ATPs are produced if it is taken in presence of oxygen, that is aerobic, and how many ATPs are produced if it is take place in absence of oxygen. So let us see from that chart, flow chart or pathway directly. See here, here this ATP is used, one ATP used, here another ATP used, two ATPs are used. Now how many ATPs are produced? If it is aerobically occur, then 6 ATP are produced plus this 2 ATP and 2 ATP. So total 6 plus 4 ATP, 10 ATPs are produced. Total 10 ATPs are produced if it is aerobic. And how many ATPs are used? 2. So out of 10, 2 are used, so net produced is 8 ATP if it is aerobic, and that is what shown on last page. Let's see. See here, if it is aerobic, total number of ATP produced 10 and total number of ATP used 2 ATP. So 10 minus 2, 8 ATP. So net production of ATP during aerobic is 8 ATP. Now let us understand how many ATP produced during anaerobic respiration. If it is occur anaerobically, then this 6 ATP will not be produced, because to produce 6 ATP, NADH2 has to transfer into mitochondria in presence of oxygen in mitochondria. NADH2 undergo oxidation to produce 6 ATP. But if it is anaerobic, means if mitochondria is there and oxygen is not there, and if mitochondria and oxygen both are not there, then this do not produce. That means in anaerobic condition, only this 2 ATP and 2 ATP, 4 ATP are produced. So during anaerobic condition, this 2 plus 2, 4 ATP produced, and this 2 plus 2, 1 plus 1, 2 ATP are used, and that is shown on last page. See, here on this last page, it is shown. In anaerobic condition, total number of ATP produced 4 and total number of ATP used 2. Thus, the net production is 2 ATP during anaerobic. So we got it. So how many number of ATP produced during aerobic respiration, or glycolysis occur during aerobic condition, or how many ATP produced during glycolysis when it occur in anaerobic condition. So here we can conclude that glycolysis, which occur in cytoplasm with the stepwise breakdown of glucose into pyruvic acid with the production of 8 ATP and 2 ATP during aerobic and anaerobic respiration respectively. Thus here the explanation about glycolysis is completed. I hope you did understand, and the notes are given in clear. So you please practice this pathway if possible to you many times till you can draw it accurately without any mistake. So practicing it will help you. Otherwise, if you got it, then explanation becomes very easy, as we have to explain step after step what is seen in that pathway. Once again I repeat it, how to explain. Glucose is the substrate, glucose 6-phosphate is the product. How to pronounce it and how to make the sentence. Glucose undergo phosphorylation in presence of ATP to produce glucose 6-phosphate. Enzymes, hexokinase. This same sentence you have to write as an explanation, sufficient. And this pathway you have to know. Question can be asked, draw a schematic flow chart of pathway of glycolysis, or draw a flow chart of EMP pathway. Question can be asked, explain balance sheet of ATP production during aerobic and anaerobic condition. There may be many questions in NEET. More than 25 to 30 best questions can be found on this glycolytic pathway. So you would come across, I will explain you simultaneously as and when we will have that video about NEET. So here some of the questions. That substrate level phosphorylation, that is inorganic phosphate combined with ADP to produce ATP, and this inorganic phosphate is available from S3PO4 to convert PGL into 1,3-DPGA. This is substrate level phosphorylation. These phosphorylations are not substrate level. This is the substrate level phosphorylation, can be asked in NEET. Moreover, which mineral act as a cofactor during glycolysis? So remember magnesium and potassium as last step that occur in presence of magnesium and potassium, which I have written here during explanation. Go through this, you will find that magnesium is written as and when it is required. And lastly, in last step, that magnesium as well as potassium is required. This is also point of NEET. Okay, I'm happy that you are always with us to see the video. You would have liked it, and moreover, if you are any doubt, you can ask in comment section. I appeal you to share and subscribe our channel, as well as keep on pressing bell icon so as to receive your new notification. Thank you. Enjoy.